So the next part is that we have a long-lasting tradition at Fysica, and that is the announcement of the prestigious Fysica Prize, followed by the presentation of the prize winner. And for this ceremony, I would like to invite Professor Piet Mulders from the board of the Stichting Fysica for giving the laudation. Piet, the floor is yours.
Okay, this is a pleasure for me to take this role, which in fact should be done by the chair of the Stichting Fysica, Art Klein, but he is in China, as many of you know, so I have the honor to tell you why we decided, or why the Fysica Prize was awarded to Marileen Dogterom. First of all, the Fysica Prize is a yearly prize, as was already mentioned, and it's given to an eminent physicist working in the Netherlands. First on Fysica. That's not Fysica as written over there with an F, but with PH, which tells you maybe something about how old the foundation is. But even that's not that old. It exists. It was established in 1948 by the Netherlands Physical Society. And the primary goal of this foundation was to foster the advancement of physics in the Netherlands, just as the goal of the society, in fact. And the foundation administers the royalties after transferring the right to use the name of the journals Fysica A to E to Elsevier's publishing company. And most of actually these royalties are used for support for the Netherlands Physical Society, but in, as I would say, the best of the Dutch economical tradition, the consecutive treasurers of the foundation have created a financial reserve from which the foundation is able to do some nice things, give real support to activities, and in particular selecting activities if they are aimed at or initiated by students or schools or universities. So it's awarded since 1986, and the decision on the Fysica Prize, and with it the obligatory Fysica lecture and a write-up in the journal, is taken by the chairs of the Fysica Foundation and the Netherlands Physical Society. But they consult representatives of the physics community, the adviesraad, the divisions of the society, the deans of the universities, directors of institutes, and previous winners. Okay, then to the recipient of this year of the prize. It's Marileen Dogterom. She's a world expert in experimental biophysics of cells. She's one of the pioneers that brought single molecule biophysics research to the Netherlands, or in the Netherlands, and stimulated it, and brought it to its present frontier position in the world. A whole new generation of physicists is presently active in this field, and you have been serving as a role model for many of them. Having worked for many years at the Vom Institute Amolf and the University of Leiden, you are now at the TU Delft, and there you are heading the Bio-Nanoscience department, working at the boundaries of nanoscience and cell biology. Besides actually those research activities, you also contribute actively in developing vision documents on the future of physics and playing a prominent role also in outreach activities. So before giving you the floor, I would like to hand you what belongs to this, and please come forward. So it is a certificate signed by the chairman and actually an envelope telling you how to get some of the, how should we call it? I phrased it here as, doe maar gewoon, dan doe je al gek genoeg. It's 2,500 euros, that's the this prize. I heard somebody told me that this is a secret. It's not a secret. And actually, these are of course simple things, but the important thing is that and Dennis Heinsberger, the secretary of the foundation, phrased it last year so beautiful. It's the perpetual and never-ending honor bestowed on you because of this prize. Congratulations.
Okay, thank you very much, Piet. Can everybody hear me well enough? Of course, many thanks to the Stichting Fysica, to the NFV, to all the people who were, without me knowing, involved in getting me here on the stage today. I feel extremely honored. And I remember, I think, Art calling me already in December and saying, are you available to give a lecture on April 10th? And I said, well, I have to check, because if you don't, we will not give you the prize. I said, okay, I'm available. That was easy. And so it's like I said, it's a huge honor. So what I will do today is use about half an hour to talk about the field. Of course, very much in a nutshell. So the physics of life, it's an established area of physics research in the Netherlands and also around the world. And it changed a lot over the last, let's say, 20 years. And I will talk a little bit about our own work, previously in Amsterdam at Amhof and now in Delft as well. But I also want to give you a glimpse for what has changed. And I think 20 years ago, this worldwide and also in the Netherlands, there was a huge push because of the possibilities that emerged to use nanotechnology or nano tools, force probes to study biological molecules, proteins and DNA at the single molecule level, and that made a huge boost. And I just want to give you a few examples of where we were 20 years ago and where that has taken us so far. Of course, there's many other examples that I'm not showing, it's just my personal taste, and I'm going to do it in a fairly superficial way. But what is very interesting and exciting to me, that all these single molecule experiments are very exciting and it's amazing what you can do, but the cell of course is a large collection of many such single molecules, and what is really still an enormous challenge and also a very exciting field to work is to really try to understand how do all these molecules, when you put them together in a bag, let's say, work together to make a living cell. So how does this collection of molecules get together and work as a machine? And it's really, we're now at the stage where the complexity of the systems that we can study in a quantitative way outside the cell or inside the cell is really at a much higher level of complexity, I mean, than it was 20 years ago. So I want to give you some examples of that as well. And then just use one or two slides, you know, to the future where may this lead us. So about the single molecule biophysics, so a very sort of example molecule that has been one of the first to be studied in the biophysics community at a single molecule level were these motor proteins. So a very well-known textbook motor protein is kinesin. It's a molecule that is made of actually two proteins, two similar proteins, a homodimer that entangles here in a helix and then has two heads, they are called. For some reason they're not called feet, but they're called heads, that move along a microtubule, which is the rails. It's a mechanical self-assembled polymer in the cell, and it can move by burning energy. So it's using ATP, chemical energy, burning it, breaking off one phosphate group from the ATP leading to an ADP, and you can use this energy not only to make this molecule move, but to transport stuff through the cell, and that can be vesicles that need to be transported to the end of your neuronal cells, but it can be many other things as well. And this protein was one of the first to be studied in a single molecule level because biologists, biochemists were able or managed to isolate these proteins and in a functional way in the early 90s of the last century. So this is a movie of the same motor. from a very well-known artist impression movie made by Harvard Media something. You can find it on their website. And this is just a short section out of the movie they made, but it shows you many other examples of molecular processes as we know them that are going on in living cells. And of course, this is way too empty. A real cell is full of molecules and you would not be able to see what's going on at the other side of the cell, but just for clarity's sake, these molecules have been left out, just to highlight some of the really intricate and fascinating processes that happen at the nanoscale. These molecules are just a few nanometers in size, but like I said in the beginning, how this all operates together to make a machine that can do so many functional things, biological functions simultaneously, is really something that we do not understand at the fundamental level yet, even though we know increasingly much about what these things look like and where they move and when they move. So if you're interested, I took this picture of the molecular motor, but there's very nice fragments also of how DNA is copied, how transport through the membrane happens, and it's very fascinating. So how about the single molecule biophysics in the 90s? So I was a PhD student at the time in Princeton, and I remember very well a talk at some day being given by Steve Block. Now, if you're in the single molecule biophysics field, you may have seen Steve Block. He's a very energetic, how would you say it, ADHD type person. And he came to talk about these experiments that he did with single kinesin molecules. And I really vividly remember it because it was very exciting. So, but he and there was his PhD student, Karel Svoboda, together with their collaborators, and managed to do for the first time is to really see the steps that a single kinesin motor takes along a microtubule outside the cell. And these steps are only 8 nanometer in size, so you cannot see them with an optical microscope. Normally, you shouldn't, because it's beyond the resolution of an optical microscope. At the same time, you cannot see them with an electron microscope, because then everything has to be static or dead or extremely cold and frozen. But they managed to do this by using an optical tweezer. So what they did is, you can read the abstract maybe, they simply asked the questions, do biological motors move with regular steps? And the end is, they built an instrument that allowed them to answer this question, and they find that kinesin moves with 8 nanometer steps. So they attach these motors to a small plastic bead of a few 100 nanometers in diameter, which you can see with an optical microscope. And biochemically, that was a big step forward. It was possible to stick a single motor protein. I copied this from the original Nature paper that they published in '93. They could put a single motor protein on a glass bead or a plastic bead, and they use an optical tweezer, which is not drawn here, but it's a focused laser beam that allows you to exert a force on this bead, together with an interferometry technique to really see the motion of this bead. So you can see the bead move with a certain velocity, and if you deprive the system of energy, then the bead will most of the time not move, and every time an ATP is burned, there will be a step. And this is one of the traces that they got, one of the first ones, where you see the position of the bead on average moving with some fantasy, some type of analysis. At really high forces, they could show that there's jumps that are very close, at least, to 8 nanometers. So this was in the 90s, and it was a big step forward. It was maybe not the first example, I'm not exactly sure. There were also other single molecules experiments on DNA, for example, going on at the same time, but it was all sort of in the 90s happening simultaneously. So then this was based on optical tweezers and force probes. Then it took another 10 years to basically observe the same process with a single molecule fluorescence technique, which is also very common and easy to do right now, but not 10 years ago. 10 years ago, this is now a science paper from 2004. There was a group of Paul Selvin together with Ron Vail, and they did not use an optical tweezer, but they put a fluorescent molecule on one of the 2 feet or head of this molecular motor. And then they observed the fluorescence of just this single molecule, again in an optical microscope. And so, of course, to make that happen, you need to have a fluorescent molecule that is stable enough so that you can observe it emit many photons. You need to do that in an experiment where there's not a lot of background fluorescence. You need to have stable dyes, good lasers, and very sensitive cameras. And somehow that all got together about 10 years after the first observation of the steps to make this experiment possible. And what the main question here was, when a motor moves, we know that it makes 8 nanometer steps, but that doesn't tell you anything about how the feet move. You can make 8 nanometer steps by doing this, or you can do it by walking like I do, and like the kinesin turns out to walk. But at the time of this experiment, that was not known, even though the movie that I showed you in the beginning that suggests this movement was already in the cell biology textbooks, which is was interesting. And fortunately turned out to be the right movie that they put in the textbooks. So what they, again, what you now see is the motion of this motor again, but you see a camera image with pixels and a fluorescent molecule that, if you look carefully, is sitting in a constant spot, and again you deprive it of ATP, and then every now and then an ATP is burned and this molecule jumps forward. You should say, okay, this is an optical microscope, it's fluorescent, so you should still not be able to resolve those steps because of the diffraction limit, but what is the key here, that you're sure that you're looking at just a single molecule, a single emitter, and even though the blob, the cloud that it makes on your detector is of course much larger than the steps you're trying to resolve, if you count enough photons at each specific position where the molecule is sitting, you can do a Gaussian fit to this profile and determine with pretty high precision where the center of this Gaussian is, and that, in an ideal case where you have unlimited numbers of photons, which of course you never do, should give you unlimited resolution, or at least a sensitivity on measuring displacements. And I'm cheating a little bit because this movie is actually not from kinesin. This is from a science paper 1 year earlier, done on another molecular motor called myosin, which makes larger steps, 36 nanometer steps. So that was actually easy to detect. But in this Science 2004 paper, they also resolved the now 16 nanometer steps that these individual feet makes, because the center of mass here makes 8 nanometer steps, but each individual foot steps by 16 nanometer each time it moves. And that's what they showed in these kind of plots. If you do the histogram of all the displacements here, you find a nice peak around 16 and not 8, which was a proof that really the motor moves by putting one foot in front of the other. So where are we now? This is again 10 years later. What the field is doing is now actually following these individual molecules with this kind of sensitivity, no longer in a very clean, isolated environment, but in real cells. So I just went to a thesis defense of Bram Vrevo in Erwin Peterman's group a few weeks ago, and he managed to follow individual motor proteins with this kind of techniques, where he can follow the fluorescence of a single emitter in a living worm, in one of the cells of a living worm. So it's in a complete organism where he can now follow the motion of a single motor protein in a much more complex environment, and you can compare the dynamics of different motors that are cooperating in a real biological process. So we've come a long way to really bring these kind of techniques to a real cell environment. And another example is this one that I like from Marileen Academiali. That's not from this year, but from 2 years ago. But these in vivo experiments are really exploding now. And she's following how these motor proteins that are hooked up to organelles in the cell have to find their way to a complex network of rails, and interested in how individual cargoes are moving from one rails to the other, finding their way in this complex network. And she's using a combination of super resolution microscopy, because in principle what I just said about being able to measure where individual molecules are with high precision, this works only if you have a limited number of fluorophores. So what you can do in a living cell where you have many, there's now new super resolution techniques where you turn molecules on and off one by one and do then this kind of statistical analysis to really get high resolution, super resolution information on structures, molecular structures in the cells. And that's what she did. So I'm not talking you through all the details, but she used this high resolution storm technique to really show where the rails are. So even though these rails are really loaded with dyes, by turning the dyes on one by one, you can do this super resolution trick and know where all the rails are, and then at the same time following these organelles with single particle tracking, so that with high resolution you can really see what the behavior of individual organelles is when they switch between two different tracks in a single cell. Okay, so this is 20 years of motor dynamics. I want to say a few things also about our own interests, which is the cytoskeleton itself. So motor proteins use the cytoskeleton. It's a network of mechanical polymers in the cells to find their way, to transport organelles, to organize the interior of the cell. But the cytoskeleton itself is also a very dynamic structure, and one of its famous roles is that it's responsible for for organizing the chromosomes during cell division. So here you see a movie of a cell that is dividing with the chromosomes in red, and here you see a still image of the same process with now the chromosomes in blue. And we have been, over those same 20 years, interested in how do these polymers, when they grow and they shrink, because they're very dynamic, how do they generate forces, and how do they then, these forces contribute to a correct positioning of this spindle in the confinement of a living cell. So what we've done, again 20 years ago or 18 years ago, for the first time we were able to measure in an isolated environment how much force a single growing microtubule can generate. So just again, the textbook information, individual microtubules are really hollow tubes made of self-assembled linear arrangements of proteins. They are not only, as I said, rails for motor proteins, but they constantly add and remove subunits from their end. And this growth and shrinkage can generate forces, which, for example, in this yeast cell are important to keep a nucleus positioned in the middle of the cell. So we started out in my group by building biophysical experiments with microfabricated barriers, where we could take a single microtubule, watch it grow into a barrier, and then measure how much force is generated by this growth. We later on also measured how much force can be generated by shrinking, because when the filament falls apart, provided it's hooked up to something, it can also generate a shrinking force, and this something, for example, can be, again, a molecular motor. Molecular motors do not only walk along microtubules, they only sit at the cell boundary. This, of course, is not the cell boundary, but this is an artist's impression of an experiment that we did to mimic this situation. So we put an active motor protein on a gold microfabricated ball. We let it interact with a filament that is falling apart, that is shrinking, microtubule, and we hook it up to a bead and use an optical tweezer to show that this shrinking filament, in combination with a motor that holds on to this filament, can generate a pulling force of several piconewton. So this is already no longer, I would say, a single molecule experiment. It's a supramolecular assembly of two components which you can reconstitute in a simple environment and really show very quantitatively how much force can be generated. We then went on to ask, okay, how do these forces contribute to the positioning of a spindle? This spindle has to find its proper orientation, especially in elongated or embryonic cells, and how do these filament forces contribute to that? And again, that sort of in the last 10 years we were able to also in vitro increase the level of complexity that we can really ask such questions. And the technique we are currently using for this, to make this artificial confinement, is a microfluidic technique. We did not invent this at all. A lot of people use it. And in short, you take a PDMS, a chip made of plastic with channels in it. And in these channels you push with a well-controlled pressure oil with lipids and also water with buffer and proteins. And this is a movie of such an experiment. You push this water phase into an oil phase through a narrow constriction, and out come very small droplets of a few tens of micrometers, typical size of cells, which are water in oil with a lipid, a monolayer in this case, of lipids around it. You can make this very many droplets of almost exactly the same size, and in these droplets you can encapsulate proteins, in our case tubulin, which then forms microtubules quite okay. So you have to proteins have the tendency to denature, so these microfluidic techniques are quite gentle on these proteins, so they remain functional if you do it right. And using this, it's a big word, but using this artificial confinement or this artificial cell-like shapes, we can now ask questions, okay, if I now have half a mitotic spindle, just a single centrosome, which radiates out microtubules that can interact with a boundary, and it can generate pushing forces but also pulling forces, because I can put molecular motors specifically on the edges of these lipids. Then I can really compare what how this centrosome self-organizes to mechanical models of the same problem. So it's simple enough that you can still, I'm not going to show you, but you can write mechanical models that provide predictions on how this centrosome will position itself depending on what the balance is between pushing and pulling forces that are generated. So we find that if you do not put molecular motors, that pushing filaments tend to destabilize the central position, and the centrosome always sits on the side. Whereas if you add molecular motors that pull on these filaments, you nicely position, just like a spindle, this centrosome in the middle of this droplet. And that's really a result of the motor proteins, which are highlighted in green here, pulling on the ends of these dynamic filaments. And you can go further, you can then also put two of these centrosomes in there and show that if there is no motors, these two repel each other, but again they are all the way pushed against the boundaries of these droplets. Whereas if you now put motors, you get a combination, or a competition between repulsion, that is purely steric between these esters, and motors trying to pull these centrosomes to the middle, and you get a nice equilibrium situation. And I'll show you two pictures, which is of course extremely simple still. It's just water with lipids around it, two centrosomes that are able to interact with the boundary and generate pulling and pushing forces, but that end up in a configuration that looks very similar, at least, to what a mitotic spindle does in a real cell. And I give this talk, show this data a lot to biologists, and of course I always have to emphasize that this is way simpler than a real spindle, and this is true. It's very static, it's very boring, it cannot respond to anything that's changing, and for this you need all these additional components that real spindles have. But it's a very nice platform where you can compare the outcome of these experiments with what you predict from measurements or calculations on the force balance in this system, and then add bottom-up components that we know biologically to be relevant one by one. Okay, so this was about the complexity that is increasing in these systems. I want to skip this and just show you two other examples of not our work, but of this same increase in complexity, or actually just one example. In the cells that I've shown you so far, where mitotic spindles are formed, these are large eukaryotic cells, and such spindles that are responsible for dividing the chromosomes do not exist in bacterial cells. But bacterial cells have also mechanisms to organize their interior, and one famous example is this oscillating protein profile that is allowing an E. coli cell or a bacterial cell to measure where its middle is. This is a picture taken in the lab of Kees Dekker, my colleague at Delft, and what you see is a protein polymer ring in red that is assembled at the exact middle of this cell, which will eventually pinch these two cells in the middle so that you get two cells. But to make sure that this polymer only assembles in the middle, there's a signaling molecule, it's called MIND, it doesn't really matter how it's called, that oscillates between the left and the right of these cells. This is a time series, time is in this direction, space is in that direction, and if you would look at the movie of this protein, it's constantly going from left to right with a period of about 30 seconds. And that means that on average this protein is never in the middle, so the cell integrates where this protein is over time, and the integrated intensity is high at the left and the right, but it's low in the middle, and this is where this protein polymer is now allowed to assemble. So this sounds simple, it's a Turing-like process, reaction diffusion, of proteins interacting with the membrane and then coming off. It's not as simple as that, and there's a lot of models, but we are now, by manipulating these cells in vivo and by reconstituting the same process outside the cell, we're really getting to also a quantitative understanding of this process. And this is, for example, what Case Lab is doing. They're forcing these bacterial cells in all kinds of different weird shapes and of different sizes, and that gives you predictions on how this oscillating profile, depending on the theory you believe, it should respond to this change in shape and size in different ways, and they can really measure now oscillatory profiles in cells that are artificially pushed in these different shapes. So that's one thing you can do. It's a way to test the different models that exist. And the other thing you can do is work of Petra Schwiele. So she's at the Max Planck Institute, a physicist also, but at the Max Planck Institute for Biochemistry in Munich. And she is reconstituting this complete system now also in artificial cells. So she uses microfabricated structures lined with lipids, and then has these min proteins oscillate in these chambers. And again, I'm just showing you the pictures, not telling you a lot of detail, but just to show that this is really possible. So the min protein, again, this is a time series, is oscillating from left to right. So this procedure, or this Turing-like behavior, can be reproduced, reconstituted with a minimum number of components. And it even has a similar effect on the ability of the FTSC polymer to assemble. So you're really reconstituting this spatial organization plus the effect it has on the polymerization behavior of the division protein in an artificial system. So these are just examples. I think they're there to show that the complexity of what we can do is increasing. It allows us much better to understand real biological problems. And then, of course, we think with all these bottom-up activities, one thing that both in Germany and also here we have as an ambition for the future is, okay, if we are able now to reconstitute some very simple essential processes of living cells, can we make in the end something that, for example, is able to do a minimal version of reproduction, which means growth and division, has a minimal version of being able to transfer information between a different generation, and does some kind of metabolic control in terms of keeping concentrations of small molecules constant. So this is not a goal because it would be cool to do this, but it's really based on the sort of conviction that really in the end, if we want to start to understand this cell as a machine, we should be able to also reconstitute it in its most simple way in an artificial system. So I think, I just wanted to show also that for this synthetic cells to happen, we will also rely on microfluidics. We can not only make droplets with microfluidic techniques, but also vesicles, and we will then have to add all these three components in these artificial vesicular systems, and I don't want to go to the details. I think what I will do is more or less leave it at that. I have one more slide that I wanted to show. So the message today is we've come a long way with single molecules, all the way to that we can now study them in detail in living cells. What we can do outside the cell has sort of reached a level of complexity that is much higher than it was 10 years ago. Combined with theoretical modeling, allows us to understand much better how the cells spatially organize themselves. And in the future, there will be developments of trying to combine all these things into some simple system that can do basic real cellular functions. It was said that there's a generations of, and this is my last slide, of physicists active in biophysics that are now just starting anew, and it's very interesting to see that many of them actually take this to a much more, even more complex biology. So I just want to mention two examples. There's Yoon Yuk, he's a new young tenure-track group leader in Delft, who is not only looking anymore with quantitative modeling at what happens inside cells, but is also looking at cell-cell communications. And this picture will not explain too much to you. The message is just that you can now control the expression of molecules inside cells using synthetic biology techniques, and that allow them to control what kind of molecules these cells secrete that allow cells to communicate. And by controlling the molecules that allow cells to communicate, you can also in a very quantitative way study how cell-cell communication is controlled and organized. Another example of this sort is what work done by Jeroen van Zon. He is also just starting group leader at AMOF. And what you see here again is a picture of a complete worm. And he's using single molecule fluorescence techniques, you just see the blobs here, to really show how cells that are close to each other in a developing or a growing worm, how they influence each other's expression level and thereby decide who is going to be this type of cell and that type of cell. It's just to emphasize that, you know, it doesn't stop at cells. All these techniques allow us to really look in a quantitative way at how cells communicate as well, and these new biophysicists are really going into that direction. So with that, I want to stop. This is a people that I'm very thankful for all the collaborations that go into this work. And the last slide is to thank the people who've been in my lab in the last years, postdocs and PhD students, because the biggest thank you is to them. And thank you very much for your attention.
Thank you very much, Marileen, for this beautiful and inspiring talk.